Hey everybody, I'm Silas, and welcome to Psilocybology. Let's expand our mind. This is a series where we talk about the research on psilocybin, the psychedelic prodrug found in magic mushrooms. If you'd like to support the show, please rate and review the podcast wherever you can, and be sure to share the podcast with anyone you know who might enjoy the content. I hope that everyone has been enjoying the holidays and is excited for 2019. Before we start with today's topic, I wanted to announce that on January 1st, I'm going to be launching the first ever biannual listener survey, so that everyone listening can provide feedback and direction for the show. Essentially, twice a year, for the month of January and for the month of July, I'm going to send out a brief anonymous online survey asking you what you think about the podcast and where you'd like to see the podcast go. To show my appreciation for your participation, at the end of the survey, you'll be asked to vote for which of 5 organizations that support either research into psilocybin or support the decriminalization, medicalization, or legalization of psilocybin should be the recipient of a $100 donation on behalf of all of you, the listeners of Psilocybology. You'll be able to find links to the survey on Twitter and Reddit, and I will also send out the link to everyone subscribed to the Psilocybology mailing list, which you can subscribe to on the website psilocybology.org. in order to stay up to date on any announcements about the podcast. And with that, let's get started. Today's episode is on 2 articles. Both papers are actually about the same exact experiment, but each presents slightly different data that came out of it. The first article is titled Mescaline, LSD, and Psilocybin: Comparison of Clinical Syndromes, Effects on Color Perception, and Biochemical Measures, published in 1962. The second article is titled Effects of Mescaline, LSD, and Psilocybin on Color Perception, published in 1963. To provide a high-level walkthrough of the experiment, this study used a within-subjects design where a total of 18 volunteers were given each of the 3 drugs, mescaline, LSD, and psilocybin. There were 2 additional volunteers, however, they did not complete all 3 drug trials for reasons that were unfortunately not specified. The order that the drugs were administered in was randomized across participants, and they were not told in what order they received the drugs until after all 3 trials were completed. They were, however, aware that they would be receiving psychedelics and knew to some extent what to expect. To minimize any cross-tolerance between drugs, each drug trial was separated by 1 week. All of the participants were left alone in a small room during each trial. However, an attendant was available if needed. I also just want to mention that most of the participants were graduate students in psychology, with varying degrees of familiarity with psychedelics. Before each drug trial, participants fasted between 8 and 12 hours. All outcome measures were taken immediately before drug administration and 2 hours post-administration. This includes the color perception tests, which we'll discuss in detail, but also includes symptom reports, questionnaires, and biochemical measures. Now that we have an overview of the study design, I want to bring up the doses that these researchers decided to use. If you remember from the last episode, the authors from that study made really no effort to use comparable doses of LSD and psilocybin. This is a problem because then you can't determine whether the differences observed are due to the primary effects of the drug itself, or simply a result of differences in drug potency. Fortunately, these researchers were familiar with the research that had already been done comparing LSD and psilocybin, and in fact they cite some of the studies we've already discussed on this podcast as providing the rationale for the doses they chose to use. What they ultimately decided on was to compare 10.5 milligrams of psilocybin per 70 kilograms of body weight, 350 milligrams of mescaline per 70 kilograms of body weight, and 70 micrograms of LSD per 70 kilograms of body weight. Relatively speaking, these are much more comparable doses than what we saw on the previous episode. The dose used for mescaline is a rather common dose, while the doses for LSD and psilocybin are a bit on the lower end, which the authors do note as a limitation. Quote, psychic symptoms were encountered less frequently than anticipated. Now, the researchers didn't just assume these were equivalent doses and call it quits. They actually did assess the number of symptoms reported after receiving each of the drugs in order to try and determine whether these drugs produced comparable experiences. In total, they recorded the presence or absence of 34 symptoms, which included both somatic symptoms, like nausea, and perceptual symptoms, like visual hallucinations. I'll include the full list in the episode transcript, which you can find on the podcast website. Looking at just the number of symptoms that at least half of the participants reported during each drug trial, 17 symptoms were reported after receiving mescaline, 10 after LSD, and 7 after psilocybin. This does seem to correspond pretty accurately to what we would expect given the slightly stronger dose of mescaline compared to that for LSD and psilocybin. Across all 3 drug trials, the most common symptoms were dizziness, weakness or fatigue, hot or cold sensations, and changes in mood. As I mentioned earlier, in addition to symptom reports and the color perception tests, the authors also examined a number of biological measures. Although I don't want to spend a ton of time going over all of them in a lot of detail, I do want to focus on 4 in particular that they found before we get into the results of the color perception tests. The 4 biochemical measures I'd like to talk about are inorganic phosphorus, free fatty acids, eosinophils, and copper oxidase. So starting with inorganic phosphorus, the researchers chose to examine this because previous studies have shown a decrease in the amount of inorganic phosphorus excreted in the urine after the administration of LSD. Phosphorus is a critical component to a number of processes in the body, such as glycolysis, or the breaking down of glucose in the body, in the excretion of ammonia from the body, and in the generation of ATP, the chemical that provides the needed energy to drive many of the processes in cells. Given its importance, there are serious health consequences to both too much and too little serum phosphorus, or phosphorus in the blood, although mild to moderate deficiency is generally well tolerated. The authors decided to measure both the amount of phosphorus excreted in the urine as well as the amount in the blood. What they found was that, in line with previous research, there was a decrease in the amount of inorganic phosphorus excreted in the urine after the administration of all 3 drugs, mescaline, LSD, and psilocybin. However, there was no change in the level of inorganic phosphorus in the blood after administration of any of the drugs, and blood levels both before and after drug administration were within the normal range for adults. This distinction is really key, because it indicates that the reduced amount that is being excreted is not either resulting in excess levels of phosphorus in the blood, or a result of a deficiency of phosphorus in the blood. This is also important because although higher blood levels of phosphorus has been associated with health problems, such as cardiovascular disease, changes in urine phosphorus were not associated with cardiovascular disease. In another study I came across that did find a link between urine phosphorus and health, it found that an increase in urine phosphorus, rather than a decrease like we see in response to our drugs of interest, was linked with chronic kidney disease. The next biochemical measure I'd like to discuss is free fatty acids. This is actually going to be the topic of next week's episode, so I wanted to make sure to introduce it here. Free fatty acids, also known as non-esterified fatty acids, are an important metabolic fuel, particularly during periods of fasting. Unfortunately, elevated levels of free fatty acids contribute to insulin resistance, and such elevated levels are often found in obese individuals. Free fatty acids inhibit insulin-mediated uptake of glucose from the blood and also appear to activate some inflammatory processes. In this study, the researchers did find an increase in the blood levels of free fatty acids after administration of all 3 drugs. This was not accompanied by an increase, however, in blood glucose. The authors of this paper suggest that this may be due to an increase in fat mobilization caused by the sympathetic stimulation of these drugs, since they all excite the nervous system. In addition to learning more about this topic during next week's episode, there's also an interesting paper published in 1964 examining how sedatives and stimulants comparatively produce changes in levels of free fatty acids. So look forward to that at some point in 2019. In addition to inorganic phosphorus and free fatty acids, the researchers also measured blood levels of eosinophils. Now, if that sounds familiar to any of you, that's because we actually discussed eosinophils in episode 6. Briefly, what they found in that paper was that psilocybin decreased eosinophil count. Eosinophils are a type of white blood cell that fight parasites and certain infections. And as I noted during the prior episode, high levels of eosinophils are associated with tissue damage and asthma. And just as they found in that prior paper, the authors of this paper also found a decrease in eosinophils after mescaline, LSD, and psilocybin administration. The last biochemical measure I want to discuss is blood levels of copper oxidase. Copper oxidases are enzymes that contain a copper center, and in mammals are also called ceruloplasmin. These enzymes are synthesized in the liver and are known to play a role in iron metabolism. The researchers chose to examine this because elevated ceruloplasmin has been found in patients with schizophrenia. I'm personally also fascinated with copper oxidase because, as we'll discuss in a paper published in 1967, copper oxidase has been put forward as the enzyme that produces the deep blue color from the degradation of psilocin. So what did these researchers find? The authors report a statistically significant but slight increase in levels of copper oxidase, but only after the administration of psilocybin. Neither LSD or mescaline produced any changes in levels of copper oxidase. Now, how this all ties together, I'm not sure, but it's incredibly stimulating to think about. I will also add this. In a paper from 1957, when a group of researchers were trying to extract a protein from the blood of patients with schizophrenia that they believed caused the disorder, they reported finding a blue substance in the extracts. Okay, now that we've gone through the biochemical measures of interest, it's finally time to discuss the main event, the effects of mescaline, LSD, and psilocybin on color perception. The authors chose to examine the effects of these drugs on color perception because they felt that one of the most interesting perceptual changes caused by these drugs were visual hallucinations that often involved changes in the way people see color. They hypothesized that color perception is enhanced by these drugs from the participation of stimuli not ordinarily eliciting color. They state that, quote, some psychotomimetics might interfere with the normal integration of experience within specific sensory modalities. They are essentially proposing a form of transitory synesthesia, where, as an example, some auditory stimuli may influence or produce visual perceptions. To test this hypothesis, they exposed participants to various stimuli that varied in terms of how much they involve color perception under normal conditions. As with the biochemical measures, the tests were conducted just before drug administration and 2 hours post-administration. In total, there were 5 tests administered. The first test they used was simply a task that involved discriminating between different hues. Specifically, they used the Farnsworth-Munsell hue discrimination test, which consists of 85 colored buttons arranged in 4 rows of graded hues from red to yellow-green to blue-green to purple-blue, and purple-blue to red. The buttons were thoroughly mixed, and the participants were asked to rearrange them in the proper order based off of their hue. They were then scored depending on how many they placed in the correct position. The second test utilized after images, which are the images that appear in one's vision even after the original image has been removed from view. Participants were asked to fixate on a 100-watt light box for a period of about 5 seconds. Then the researchers used a series of slides to change the color of the light. After the 5 seconds were done, the participants were instructed to close their eyes and report what color they saw, and when the after image was completely faded. Once the after image disappeared, they were then asked to match the color they were shown using a color comparator. Under normal conditions, after staring at a colored light for a set period of time, when you close your eyes, it is typical that you see the same color for a brief period before it transitions to the negative after image, meaning that you start to see the color complementary to the light you are looking at. The third test utilized what's called an episcodister. The simplest way to describe this device is that it's a spinning wheel, half of which was white and half black. Now, it looks like there were a few different types of this device, and they don't provide a ton of detail into what it really looked like, but I believe, based on some research, that the one they used had a stationary cover over the wheel that only revealed a portion of the black and white disc. As the black and white disc spun, it would give the appearance of a flicker between black and white, which can be adjusted by varying the speed of the rotation. This was used as a rather simple visual stimulus that ideally does not produce much color perception. Participants were asked to then report if the flicker evoked any color perception and to identify the color that they saw. The fourth test simply involved participants closing their eyes and listening to a pure tone, which was varied by frequency and loudness, although they report the results in aggregate. The fifth and final test combined the third and the fourth tests, whereby participants were played the pure tone while also looking at the flicker from the episcodister. Starting with the less exciting but still methodologically important results, the researchers noted that improvement in color discrimination was seen after the first and second drug trials, which they believe was due to the greater practice participants had each time they performed the task. Importantly, as you remember, they randomized the order of the drugs given to participants, so this bias should be evenly distributed and should then not impair our ability to assess whether the drugs altered their ability to discriminate hues. When it came to the color discrimination task, where participants had to arrange colored buttons in the correct gradient order, the researchers found significantly higher number of overall errors after receiving psilocybin. After LSD, participants only appeared to have greater difficulty discriminating between reds, yellows, and oranges. No statistically significant impairment was seen after being given mescaline. After the afterimage task, all three drugs improved the participants' ability to correctly match the afterimage they saw using the color comparator. Although notably only psilocybin seemed to increase the duration of the afterimages. By far the more interesting findings to me are the ones where the tests didn't have a correct response. For example, all three drugs significantly increased the perception of subjective colors after looking at the black and white flickering episcodister. What was particularly fascinating about the results from this test is that they correlated with the results of the hue discrimination test. What I mean by this is that the color ranges that they were more likely to have difficulty arranging by hue in the discrimination test were the colors they were the most likely to see in response to staring at the flickering episcodister. So for example, if an individual had a lot of trouble arranging the red and yellow shades during the discrimination test, they were then more likely to report seeing red-yellow colors during the flicker test. Now, I believe the flicker test always came after the hue discrimination task, so it's possible the first influenced the latter, but that'd have to be tested to be known for sure. It's also possible that there is an underlying reason that this particular color spectrum is implicated in both of these perception tests. For the fourth test, when participants simply listened to a pure tone with their eyes closed, both LSD and mescaline seemed to increase the number of colors participants reported experiencing. Psilocybin did not increase such experiences of color in response to the tone. Finally, when the tone and the flicker tests were combined, only LSD increased the number of color experiences reported by subjects. So overall, the researchers conclude that the results of the study tend to confirm the impression that LSD and mescaline produce more vivid visual hallucinations compared to psilocybin. Although they don't really discuss it too much, which might have been intentional, while the results were interesting, I don't believe they support their initial hypothesis, particularly given that only LSD produced an increase in color perception in the combined visual and auditory stimulus task. Importantly, the increase in color perception during this task was also much smaller than the increase seen after LSD administration in the task involving just the flicker, without the tone. If visual perception was being enhanced by the input from additional non-visual stimuli, I think then one would expect to see the greatest increase in color perception during this combined task. That said, when participants were only listening to the pure tone, they did experience an increase in subjective color perception, so clearly at least something is going on where non-visual stimuli are influencing visual perception. It just does not appear to be in the way the authors may have initially hypothesized. Hopefully someone continues to examine the effects of these drugs on color perception. A fun follow-up that I think would be interesting would be to examine how different genres of music, rather than just a single tone, might alter color perception while on psilocybin. Anyway, that's it for today's episode. As always, I really hope you enjoyed the show. If you have any questions, feedback, or ideas for the podcast, please let me know. You can find out all the ways to reach me on the website, psilocybology.org, where you can also find the full transcript for each episode if interested. Thank you all for listening, and I'll talk to you next time.